Assalamu alaikum wa rahmatullahi wa barakatuh. Today we will talk about a new chapter of pathology after the cell injury, after the chapter of cell injury. It is inflammation, including acute and chronic inflammation. An inflammatory process or inflammation is a protective response intended to eliminate the cause of cell injury as well as the necrotic debris resulting from these causes or original insult. It then heal and reconstitute or regenerate the site of injury or the site of affected site or damaged tissue resulted from this inflammatory process. Although inflammation helps clear infectious and along with repair, both inflammation and repair have considerable potential to cause harm to affected tissue or to other tissue. The inflammatory process and association with repairing process are also may induce harmful or damaged, even they are help in clearing of infectious agent or other causative agent and repairing of the affected site. On the other hand, may cause damage for other health tissue. For example, inflammation in the peritoneum leading to fibrous bands cause intestinal obstruction resulting from these fibrous bands, or pericardial inflammation resulting in a dense encasing scar that impair cardiac function or cardiac muscle movement. Due to this scar يكون non-functional non-contractile نتيجة healing by replacement the normal myocardial cell with the scar. The inflammatory players include different type of components, including circulating cells, including neutrophil, eosinophil, basophil, lymphocytes, monocyte, platelets, and also plasma protein, including clotting factors, kininogen, and complement components. Also vascular wall cells, including endothelial cells, smooth muscle cells, as well as extracellular matrix involving structural proteins, for example collagen and elastin, gel-forming proteoglycan, and adhesive glycoproteins, for instance fibronectin, that are the cell to extracellular matrix and extracellular matrix to extracellular matrix connector, اللي هو المسؤول عن الربط والconnection between cell and extracellular matrix and extracellular matrix with other extracellular matrix. The connective tissue cells including sentinel cells أو الخلايا الحارسة أو الحامية اللي هي resident cells normally present in different tissue distributed in different tissue يسمونها sentinel cells الخلايا الحارسة أو الحامية, such as mast cells, macrophages, lymphocytes, in addition to fibroblasts that synthesize the extracellular matrix and can proliferate to fill in a wound. All of these players previously mentioned or mentioned above, including circulating cell, plasma protein, vascular wall cell, extracellular matrix, connective tissue cells. All of these players interact together to resolve a local injury and restore normal tissue structure and function, as much as not necessary. This figure shows the components of inflammatory process, or many players of inflammatory process, including vascular wall cells or circulating cells, including polymorphonuclear cells اللي هي نفس النيوتروفيلز. الفانكشن مالتها to eliminate, its act to eliminate of microbes and dead tissue. Also lymphocyte, which considered as main immune cells that responsible to produce different type of cytokines and other chemical mediators. Also the mast cells, which considered as resident cells in connective tissue, and it is main source of vasoactive amines such as histamines and other mediators. The monocytes, which named monocyte in the blood vessel, it is called monocyte. In tissue or in extracellular tissue or in interstitial tissue is called macrophages, and its role is elimination of microbes and dead tissue, source of mediators. It is a source of mediators such as cytokines and other mediators, and it has other immune responses. Also the cells of wall, blood vessel wall, including smooth muscle cells and endothelial cells and basement membrane. Endothelial cells considered source of mediators such as nitric oxide, cytokines and others. Also the plasma proteins, including complement and clotting factor. Complement act as mediators of inflammation and help in elimination of microbes. Clotting factor and kininogen act as mediators of inflammatory process. Also as well as the extracellular matrix, including fibroblast cells, other collagen and elastic fibers and fibronectin adhesive molecules, also play an important role in repairing process, particularly the fibroblast, which is responsible to produce this extracellular matrix and filling the affected area of wounds. The inflammation include or occur as two types of inflammatory process, either acute or chronic inflammation. Acute inflammation is of relatively short duration, lasting from a few minutes up to a few days, and is characterized by fluid and plasma protein exudate and a predominantly neutrophilic leukocyte accumulation. In contrast, the chronic inflammation is of long duration, continue for days to years, and is typified by influx of lymphocytes instead of neutrophil in acute, and lymphocyte and macrophage, with associated vascular proliferation and scarring. Vascular proliferation and scarring means initiation or start with a repairing process. Usually the chronic inflammation, and with initiation of a repairing process or healing of the affected site. Acute inflammation, this process has two major components or events. Vascular changes and cellular events. Vascular change means alteration in vessel caliber resulting in increased blood flow, vasodilation, and increased blood flow to affected area, and increased vascular permeability. Increase vessel caliber or diameter resulting in increased blood flow, vasodilation, and increase vascular permeability that permit plasma protein and the fluid to leave the circulation into interstitial tissue. The cellular events means a migration of leukocytes from microcirculation, particularly the venule, post-capillary venules, into the focus of injury. It means cellular recruitment and activation. The 5 classic cardinal signs of acute inflammation are heat, color, طبعا هي of affected site, not of the whole body. Cardinal signs of injured or inflamed site. Acutely inflamed site are heat, color, not the fever, not means the fever, يعني localized heat of affected site. A redness due to redness or erythema, rubber due to vasodilation and increased blood flow, as well as swelling or tumor due to extravasation of fluid, protein-rich fluid, resulting in swelling of affected area. Pain or dullness due to mediated by different chemical mediators or may due to compression of the swelling resulting from collection of edematous fluid on nerve ending, and also loss of function, loss of function of affected site. Vascular change after a transient or second vasoconstriction, arterial vasodilation occur. يعني in early stage there is a vasoconstriction, يعني sequence of vasoconstriction, and then the arterioles will undergo vasodilation, resulting in locally increased blood flow and engorgement of capillary beds with the blood, causing redness or erythema and warmth. نتيجة الفريكشن between blood and increased blood flow or fast blood flow with vessel wall يصير الورم. Consequently, the microvasculature become more permeable, resulting in leakage of protein-rich fluid into extravascular tissue, causing concentration red blood cells, increasing blood viscosity, and the slowing of circulation or stasis. يعني بعد ما يصير extravasation of plasma fluid or protein-rich fluid from the intranal vessels into يعني adjacent tissue, extravascular tissue, the RBC will become concentrated inside this blood vessels, and concentrated blood RBCs will result in increase blood viscosity, and the blood viscosity will lead to slowing of circulation or stasis. Then leukocytes, principally neutrophils at the early stage, will settle out تنفلت يعني of the flowing blood and accumulate along the vascular endothelial surface. لأنه بالنورمال احنا البلاد فلو يكون على شكل اللامينار فلو، الاكزيال كارنت مالت the center تكون الار بي سيز and then the white B cells, and then a thin layer of plasma separate the blood component from the direct contact with the vascular endothelial cells. فلما يصير blood stasis نتيجة extravasation of fluid and concentrated blood و increased viscosity راح يصير stasis يعني sluggish blood flow. فالwhite BCs will settle out toward يعني move toward the blood or toward the vascular endothelial surface, تكون directly contact with endothelial cells اللي يسمونها process what we call it margination. This is the second step. The first step is the stasis and then margination. Then the leukocyte adhere to endothelial cells because it is become direct contact with endothelial cells, and sequence between them, between يعني endothelial cells, and migrate through the vascular wall into interstitial tissue, migrate through the gaps between the endothelial cells and penetrate the basement membrane and exit into affected site. There are some pathological terms, transudates and exudates. Transudates means an extravasation of blood plasma containing little proteins. Extravasation of blood plasma containing little proteins. While the exudate, which mostly seen in inflammation. Exudate mostly associated with inflammatory process means extravasation of protein-rich fluid. And even cells, inflammatory cells, into the interstitium. This fluid accumulate is called edema. يعني الإكزودات هي inflammatory edema. Transudate هي not inflammatory, maybe not inflammatory edema. This figure shows the capillary or vascular network in inflamed area compared with normal one in normal area vascular network, where this within normal limits, physiological limits, normal size, normal diameter, and normal fluids in interstitial tissue, normal cells. This is represent only sentinel cells or resident cells, الخلايا اللي هي normally present as sentinels يعني الحوارس أو الخلايا الحامية. While at the inflammatory area or inflamed area, the vascular bed will undergo dilation, vascular dilation, including arterial dilation and expansion of capillary beds, and venular dilation, resulting in arterial dilation, resulting in increased blood flow to site of inflammation and giving the area a red color. Also, the increased dilation of venules, resulting in extra or protein-rich fluid, leakage of plasma protein or protein-rich fluid into interstitial tissue, mostly associated with leakage of inflammatory cells, particularly the neutrophil in the early stage forming exudate or inflammatory exudate, resulting in swelling of affected area. Endothelial cell contraction leading to intercellular gaps. Endothelial cell contraction leading to intercellular gaps. Increase intercellular gaps between endothelial cells and venules and is a reversible process elicited rapidly by binding of histamine, bradykinin, leukotrienes, and other chemical mediators to specific receptor. The main sites for this rapid increase in vascular permeability are post-capillary venules. Post-capillary venules may be due to expression of more number of receptors for this chemical mediators compared with other vascular bed. Endothelial cell retraction, endothelial cell contraction, and endothelial cell retraction is another reversible mechanism resulting in increased vascular permeability by effect of tumor necrosis factor and interleukin-1. Also increase or amplify the vascular permeability, but mediated by other chemical mediators. They are tumor necrosis factor and interleukin-1. Sometimes the endothelial cells directly affected by injurious stimulus or severe insults. Direct endothelial injury results in vascular leakage by causing a direct endothelial cell necrosis and detachment. This effect is usually seen after severe injury such as burns or infections during produce their toxins, severe toxic secondary metabolites of infectious agents will result in a direct injury or direct necrosis and detachment of endothelial cells, resulting in increased vascular leakage of protein-rich fluid to interstitial tissue. This figure shows the steps or types of extravasated fluid compared with normal one. In the normal state, the hydrostatic pressure within normal limits and colloid hydrostatic pressure maintained by sodium salt content in the blood or sodium concentration in the blood, and it is directed against the blood vessel wall, while the colloid osmotic pressure maintained by concentration or plasma protein content in the blood, particularly the albumin. This is maintained within normal physiological limit in normal state. If there is an increase in hydrostatic pressure, such as in case of venous outflow obstruction or congestive heart failure, this pathological cases will lead to increased hydrostatic pressure. Increased hydrostatic pressure will result in a leakage of fluid into interstitial tissue. Protein poor fluid. Also decrease in colloid osmotic pressure due to decrease in protein synthesis, such as for instance in the liver diseases or increased protein loss, such as proteinuria in kidney diseases. This will result in decreased osmotic pressure due to decreased protein blood protein or plasma protein content and this will result in extravasation of fluid from the vessel into interstitial tissue. But the fluid is protein poor fluid. This is represent a transudate. Transudate means extravasation of protein poor fluid into interstitial tissue. In the inflammatory process, increased vascular permeability and vasodilation and increased vascular permeability due to contraction and retraction of endothelial cells and increase the gaps between them. This will result in increased leakage of protein-rich fluid from the vessels into interstitial tissue, and sometimes containing or rich with inflammatory cells, and this represent exudate or inflammatory exudate. This is the differences between the exudate and between the transudate and exudate. cellular events. احنا اخذنا vascular events as cellular events. The sequence of events in extravasation of leukocytes from the vascular lumen to extravascular spaces is divided into different steps. Margination and rolling. Margination means the leukocyte move toward or settle out toward the endothelial surface, vascular endothelial surface. Margination and rolling of leukocyte on endothelial surface. Adhesion and transmigration of leukocyte between endothelial cells, and migration in interstitial tissue toward chemotactic stimulus. Chemotactic stimulus or chemotactic mediators. Chemical mediators. It is a chemical mediators act to recruit leukocyte to site of injury or to site of inflammation. After transmigration of leukocyte through the endothelial cells and basement membrane. Rolling, adhesion and transmigration are mediated by the binding of complementary adhesion molecules on leukocyte, expressed on leukocyte surface and endothelial surface. Rolling, adhesion and transmigration are steps of extravasation of leukocytes mediated by these complementary adhesion molecules. Molecules expressed on the surface of leukocyte and endothelial cells. They see interaction between them as we explain that. This table only يعني للإطلاع الانdothelial and leukocyte adhesion molecules. هاي اللي هي molecules which mediate the interaction and adhesion of leukocyte through transmigration or extravasation of leukocyte to interstitial tissue. مثلا, for example, P-selectin expressed on endothelial cells will combine or binding with sialyl Lewis X modified protein, and this help in rolling of neutrophil monocyte and lymphocyte rolling. مثلا عندنا L-selectin on endothelial cells combined with sialyl Lewis X modified protein and help in rolling and adhesion neutrophil monocyte and T lymphocytes Also intercellular adhesion molecules اللي هو considered as immunoglobulin superfamily interact with integrins and help in adhesion, arrest, transmigration of neutrophil monocyte and lymphocyte. This is important adhesion molecules that mediated the interaction between endothelial cell and leukocyte through the transmigration and extravasation of leukocyte to the site of inflammation in order to clear and remove the injurious stimulus or الكوزج يعني نقصد. Margination and rolling. The smaller discoid red blood cells tend to move faster than the larger اللي هي الليكوسايتس الأكبر spherical leukocyte cells. Therefore the leukocytes are pushed out of the central axial column لأن بالcentral axial column of blood flow حتكون ال RBCs بعدين تجي وراها ال white blood cells where they normally flow and accumulate and interact with endothelial cells يعني من يصير settle out from the center لأنه البلاد النورمال بلاد فلو يكون على شكل لامينار زين فمن يصير stasis مثل ما حكينا قبل شوية راح يصير sluggish blood flow فالوايت blood cell will settle out or move toward the endothelial cells. هاي المرحلة يسمونها المارجينيشن. Subsequently, leukocytes tumble on the endothelial surface and transiently sticking along the way, a process called rolling. أول ما تجي يسوي يعني يصير interaction between endothelial cell and leukocyte and mediated by loss adhesion by connection of E-selectin and B-selectin on endothelial cells and L-selectin expressed on leukocyte. The relatively loss and the transient adhesion involved in rolling are accounted for by the selectin family. قلنا ال L-selectin على الليكوسايت وال B-selectin وال E-selectin على الاندوثيليال سيلز راح يصير بيناتهم interaction يسمونها هاي مرحلة يعني used by leukocyte in loss adhesion مرحلة loss adhesion to help the leukocyte in rolling process حتى يسهل عملية الرول. Selectin are receptor expressed on leukocyte and endothelial cells. These include E-selectin اللي هو CD62E confined to endothelial cells, B-selectin على endothelial cells والبليتلت, والL-selectin مثل ما قلنا on the surface expressed on the surface of leukocyte. The endothelial selectins are typically expressed at low level or are not present at all on normal cells. يعني بالnormal state selectins are not expressed on normal endothelial cell, but if there is an injury or injurious stimulus will stimulate the production of specific chemical mediators, and this chemical mediators will stimulate endothelial cells to produce or express this selectin on their surface. Adhesion and transmigration. عندنا الأولى margination and rolling. and then adhesion and transmigration. Eventually, the leukocyte can firmly stick to endothelial surface. الفيرست ستيج كان loss stick، يعني الاديجن between endothelial cells and leukocyte يكون loss or weak حتى يساعد الليوكوسايت to roll on the endothelial surface. In this stage, the adhesion will convert to firm adhesion or firm stick to endothelial cells, endothelial surface اللي يسمونها مرحلة الادhesion. This firm adhesion is mediated by other adhesion molecules، يعني مو السلكتينز اللي كانت باللوس أو الويك adhesion، لا other mediators or other adhesion molecules represented by immunoglobulin superfamily مثل ما قبل شوي بالتيبل حكيناها، اللي هي من ضمنها الانتر cellular adhesion molecule 1 and vascular cell adhesion molecule 1 on endothelial cell that interact with integrins اللي هي glycoproteins expressed on leukocytes cell surfaces. راح يصير interaction between these integrins اللي هي glycoprotein and this immunoglobulin superfamily mediating the firm adhesion between leukocyte and endothelial cells to help the extravasation of leukocyte into inflamed area. Integrins are normally expressed on leukocyte plasma membrane but do not adhere to their appropriate ligands until the leukocytes are activated by chemotactic agent. يعني هو normally present موجود على leukocyte بnormal state but not adhere to specific ligands إلا بعد ما يصير activation للليوكوسايت by chemotactic agent. chemotactic اللي هي الكيميكال mediators اللي يسوي recruitment، يعني استدعاء للليوكوسايت to affected site or to site of causes. High chemotactic agent or other stimuli produced by endothelium or other cells at site of injury. This figure shows the steps of extravasation of leukocyte. Here, for example, neutrophils, extravasation of neutrophils into an inflamed area or into site of causative agent. The initiation of inflammatory process or transmigration of leukocytes start from the causes, for example infectious agent or others, engulfed by resident macrophage. قلنا اكو resident macrophage يدخل اكو فد causes or infectious agent to interstitial tissue، فراح يلقى الماكروفاج resident or sentinel macrophage and engulfed by macrophages. Then activated macrophages will produce different cytokines including tumor necrosis factor, interleukin-1, and this will stimulate the endothelial cell to express their receptors and adhesive molecules مثل البي سيلكتين والإي سيلكتين. Also the leukocyte marginate toward the endothelial cells and express the adhesive molecules CLI, Lewis X modified glycoprotein and integrins, but the integrins in the early stage يكون has low affinity. Then the leukocyte connect or bind with the endothelial cells using selectins. B and E on endothelial cells and L-selectin on leukocyte at the stage of loss or weak adhesion to help the neutrophil to be rolled on the surface of endothelial cells until reach the firm or the site of firm adhesion, which mediated by other molecules اللي هي قلنا immunoglobulin superfamily مثل intercellular adhesion molecules, cell adhesion molecules, vascular cell adhesion molecules expressed on endothelial cells, and integrins اللي هي glycoprotein expressed on neutrophils. Also the transmigration of leukocyte through the interendothelial gaps mediated by platelet endothelial cell adhesion molecules, and then transmigrate through the endothelial surface and penetrate the basement membrane and extravasate this neutrophils into site of injury or into site of causes recruited by chemokinase or chemotactic mediators, and then face the causative agent and engulf and remove this initial insult. اللي هنا كافي. إن شاء الله نكمل المحاضرة الجاية. السلام عليكم ورحمة الله وبركاته.